A very good day, everyone. So today is my utmost pleasure to welcome Professor Zhang Hao as a speaker in the CBC Virtual Seminar Series. So Professor Zhang is a professor in the Department of Biomedical Engineering and Department of Ophthalmology in Northwestern University. He is also a co-founder of Optician Incorporation, a company that is part of the medical equipment and supplies manufacturing industry located in Illinois. So Professor Zhang completed his undergraduate degree in instrumentation engineering and computer science from Shanghai Jiao Tong University in China in 1997, his master's in biomedical engineering from the same university, and his PhD in the same field from Texas A&M University in 2006. So after his postdoctoral training in the area of photoacoustic microscopy with Professor Wang Lihong at Washington University in St. Louis in 2007, he briefly started his faculty appointment at Department of Electrical Engineering and Computer Science at the University of Wisconsin-Milwaukee before moving to the Department of Biomedical Engineering and the Department of Ophthalmology at Northwestern University, where he has been there ever since 2011. And Professor Zhang has won numerous awards throughout his career, and to name just a few, the NSF Career Award, NIH Director's Challenge Award, NIH Impact Award, SPIE Translational Research Award, and the US National Academy of Sciences Kawasaki Prize. Professor Zhang has performed pioneering works in the field of photoacoustic microscopy and super resolution optical imaging. And his current research focuses on optical coherence tomography for eye and brain, and also single molecule localization microscopy, technology development, and applications. So without further ado, let us welcome Professor Zhang, presenting the topic on spectroscopic single molecule localization microscopy. So Professor Zhang, please.
Thank you. It's a pleasure. Let me share my screen. Thank you very much for the kind introduction. It's a pleasure to share my work to colleagues at, far away from here. And see whether we can find common ground and to start possible collaborations. I'm a professor at Northwestern University. My major appointment is with biomedical engineering. I have a courtesy appointment with ophthalmology where I do most of my clinical work. Today's topic is called spectroscopy. It's called spectroscopy super resolution. And then the super resolution technology we're using is called single molecule localization microscopy. So, one slide to show my overarching research goal. So my lab focuses on vision research or ophthalmology. We are developing technologies trying to address blinding diseases or vision science. We develop tools to study from human to small animal to single cell to single molecule. So the overall theme is trying to find ways to better understand and better diagnose blinding diseases like glaucoma, macular degeneration, or other type of diseases. So we have two technologies, OCT and super resolution. Today I will focus on super resolution only. So the super resolution technology became more widely accepted and widely recognized after the 2014 Nobel Prize in Chemistry. The super comes from, so if we look at this curve is called AB disk, is limited by the wavelength and numerical aperture. Essentially, there is a ultra limit for optical microscopy to see the size of an object. It's fundamentally limited by the wavelength, by the lens parameter, which is translated to be to the numerical aperture and scaled by a coefficient. How to overcome this becomes a major challenge for researchers to spend their time and effort trying to overcome. This value, rule of thumb, this value is on the order of 200 to 400 nanometers. So how to go to single digit nanometer has been impossible until 3 groups of professors published their independent research. So I showed 2 groups. One is called stimulated emission depletion microscopy. It's called STAT. It was developed by a researcher from Germany. He was born and raised in Romania, and first published that result when he was a professor in Turku, University of Turku, 3 hours away, 3 hours east to Helsinki in Finland. His name is Stefan Hill. In German, people pronounce Stefan Hill. So the other group of technology that awarded the Nobel Prize, not everybody awarded, but Eric Benzik awarded Nobel Prize. I would like to recognize all the 3 researchers. They contributed significantly to the technology. It's called a single molecule localization microscopy. Eric Benzik and Howard Hess, they were co-authors when they built their first prototype in Eric's father's living room. And almost at the same time, slightly later, the professor Xiaowei Zhang from Harvard published a similar technology, not entirely the same in chemistry, but the algorithms are highly similar. It's called a storm. Both of them trying to detect a group of photon and try so that they can manipulate the photon emission to be statistically from single molecule, and they can ignore the Poinsett function. So what is storm or POM exactly? What's the underlying principle of the single molecule localization microscopy? Let's look at the first figure. There are 5 molecules. They're so close to each other because of the optical diffraction limit. When you have an objective lens using confocal or epifluorescence microscopes, they are 5, but from the image, you won't be able to tell how many they are. You will see a blur. If there's a magic technology that we can only allow 1 of the 5 emit fluorescence photon at a time, all right? Only 1 molecule at a time. So that as long as I know statistically there's only 1 molecule, I can basically ignore the size of my Poinsett function. I just find the Poinsett function, I find the center, and that's my location of the molecule. And then I repeat the process 5 times. I will have an image. So this is a process for localization, ignoring the size of the Poinsett function and find the center. This is the numerical localization. Repeat the process thousands of times, we will have an image. So this is the fundamental process of single molecule localization microscopy. On the left, you will see a fluorescence microscopes under the diffraction limit and the super resolution beyond the diffraction limit. That's the super resolution, the super part. These are all good and then awarded Nobel Prize. What are we doing? So if these 5 molecules are different, how can we solve them? How can we recognize them? I mean, people recognize this challenge a long time ago. And then one of the most straightforward way is we will label these 5 molecules using dyes that are far away, so that their molecular emission spectrums are far away. I can use diquark mirrors to separate them. So people introduce a name called channels or bands. But in terms of how many bands you can get, as many as 4, that's the maximum people have been showing. Why that's the case? The complexity of the system and the photon budget. As long as you have more component in your system, you have reduced photon budget. What if the fluorescence label have overlapping emission spectrums? How can you use anything to differentiate them? So we might need a better tool to solve this challenge. And I'm sure some of you may find this similar. So when you have a light coming, we can actually potentially put a dispersion component. And this dispersion component will be able to separate the light into 2 parts. The one part carries the original spatial information. The other part carries the spectral information. How to implement the dispersion component? Different groups have different methods. So one of your idea was to use a beam splitter plus a prism. So that's actually very smart. And we use a different alternative is to use a prism. So when we insert a prism, the prism has two orders of diffraction, zeroth order and first order. For most people using grating, for most people using grating, the zeroth order was trying to suppress so that all photon goes to the first order because people care about the spectrum. But if we manipulate the grating carefully enough, we can control the splitting ratio between zeros and first order so that certain photon will form the spatial image, go through all the localization process. And certain photon will have corresponding color dispersion per blinking. So these two images have to be acquired at the same time in order to satisfy the stochastic process need. And then based on the location and the color, we can reconstruct the image just by routing all the photons into the single camera. So this has to be acquired simultaneously, or you have to use two cameras and synchronize them. So we use one camera because it's easier to implement and then it's less alignment. So this technology was primarily came around 2015 to 2016. So three groups published the first wave of paper. Xu's group from Berkeley. There is a group from Cambridge in our group. I think we are the second. The Cambridge group was the last. I have to say, we know the best on optics, but we really don't know how to do the cell preparation or the staining. So that's give us a big headache. It's a learning process for us. This shows the real optical setup on the right side. We use a Nikon inverted microscope body, and inside the box, that's the grating and the merging optics. And we use a EMCCD to acquire everything. So on the lower left side, you see the zeroth order frame and the first order frame. Zeroth order frame, you see the blinking like any storm that you will acquire. And these blinkings give us the location of each single molecule. And on the right side, that's the dispersion corresponding to each blinking. We have to do further analysis to get the spectroscopy information out of each first order image. So I'll show how this is done. So I'll give you an example. This is, if we don't have spatial separation, if we don't have spectral separation, we're using this, they're highly overlapping. They're almost the same if we don't have the optimal spectral analysis. So look at this point cloud or pixel cloud. Without color separation, we may highly likely to conclude that this is a single molecule, and all these photons and all this localization will be combined into a single one. But with what we have as the first order dispersion, we are able to separate, no. They're actually two molecules. They're so close to each other, but they have totally different emission spectrums. This is how they look like. There's one molecule, a second molecule. If we are unable to separate the spectrum emission, they will look like one. Of course, this is an extreme case. When we label the real cell sample, so we pick these three colors to show. Primarily, we pick two, the Alexa fluor and mito. They're almost the same in terms of emission spectrums with tiny difference. If we don't have the whole spectrum, it is basically impossible to separate them. But because we have the capability to analyze the whole emission spectrum, we not only separate them, we separate them from the autofluorescence. I'll come back to this. What is the autofluorescence and how we call that? There's a special name, people call this fluorescence impurity. We have a whole study to investigate how to get rid of the fluorescence impurity. So what's the indication out of this study? The number of different labels is essentially unlimited. Why that's the case? As long as we can numerically analyze their full spectrum, we don't impose any requirement for the optics. We acquire everything and we numerically analyze them, subjected to signal-noise ratio. As long as we can acquire high quality spectrums, we don't need any optical differentiation to separate molecules. So what we do with it? one of the study was trying to image mitochondria under certain pathological conditions. Because this is recorded, so I didn't show all the ongoing studies. So the result may seem a little bit discontinued. So one of the major study we're trying to do here is we harvest retinal cells and different retinal cells, and then we subject the retinal cells to glucose at different time duration. How these cells adapted to high glucose have significant indication in diabetic retinopathy in patient. So our solution is to A, image the mitochondria, B, isolate different mitochondria, C, quantify the mitochondria morphology, and then how to separate the morphology on the different glucose condition. We're trying to expand the glucose to high intraocular pressure as well. So come back to the spectral analysis. As I mentioned here, the number of different label is in theory unlimited, but there's a catch. The signal noise ratio has to be sufficiently high so that we can get the smooth spectroscopy analysis. In most cases, this is what we get. There's just lots of noise because photon is always limited. For a good fluorescence label, you got 5,000, that's already very lucky. And after we divided them into 2 channels, 3 to 7, 4 to 6, or 5 to 5, we have even less photons. And when you have all the radar noise, all the thermal noise of the electronics, all spectrums will look like this. So let's say we select these 2 dyes, CF647 and then AF647 and CF660. The spectrum looks similar. If we do not have the right way to fit the profile, we may have crosstalks. In other words, if we plot the distribution of the spectral centroid, you see the overlapping area. So for any inaccurate classification, we may misidentify the molecules to a different dye. This is how the point cloud look like. They're really close. Using machine learning. So if we train everything carefully, and the machine learning algorithm will be able to tell from this noisy data. So in other words, if we do just filtering, you will see the background a lot more unspecific staining or crosstalks. And after machine learning, the background became a lot more cleaner. Of course, we're still unable to achieve 100% cleaned. So with the help of multicolor and machine learning, we're really trying to push the super resolution to retinal diseases. One of the study is to create a high pressure model in the eye, and then we harvest the cornea to investigate how different drugs and how the high intraocular pressure affects the behavior of the cellular structure and how the drug will potentially reverse that. This is a working manuscript, so sorry, I won't show more details about this work. So let me focus more on 3 of the technologies that we have been focusing on. One of that is, if you recall, I mentioned that by separating the beams into first order and 0th order, we have 2 beams to begin with. One of the challenges is how can we do 3D image? There are 2 major ways to do 3D imaging in single molecule localization microscope. On the left, this is called a points per function engineered or astigmatism-based super resolution. In other words, researchers introduce a cylindrical lens. Because of the cylindrical lens, the points per function will become dispersed along either X or Y axis with respect to the Z location. So we can analyze the distortion of the points per function with respect to the either X or Y to back calculate the Z. The other method is called the dual plane. The dual plane method is if we separate the beam, separate the detection into two arms, one focus on one imaging plane, the other focus on different imaging plane, we may have two calibration curves. These two calibration curves can be collectively used to localize the Z location. So this is called a biplane. We are using biplane to achieve 2D, 3D imaging in spectroscopy single molecular localization. So I show the overall architecture again. The starting point is the fluorescence emission. We have an imaging lens, a dispersion component. Dispersion component is a grating, and in this case, so we have two beams, spatial beam and spectral beam. Pay attention here is we have two beams already. So one of the easy part is we just add a pair of mirror to one of the beam. In this case, we add that to the spectral beam to introduce a optical pathless difference. In other words, we artificially move one of the imaging plane to a different location with respect to the spatial location of the first zeroth order beam. So how it look like? The gray plane is the real original imaging plane. Now we have the spatial imaging plane slightly shift forward. We have the spectral imaging plane slightly shift backward. What does it give us? We will be able to manipulate the point spread function with respect to the distortion along one direction. I have to make it clear that we cannot analyze on both directions because we have a spectral dispersion to begin with. So we can only class everything along one direction and compare the distortion along only one direction. In this case, it's X direction. So what we did is we put a quantum dot. We shift from -700 nano to +700 nano. Measure the zeroth order image, the first order image with respect to different Z depths to obtain these two calibration curves. These two U curves give us the back calculation of the Z location. We have two beams to begin with. One is for spatial imaging, the other one is for spectral image. And we introduce a artificially path delay. We have a 3D imaging by itself. We don't have to introduce the dispersion component. Sorry, the distortion component like the cylindrical lens. So this is a demonstration of a cell image from 1, 2, 3 different slabs. One of the major benefits for biplane as compared to the astigmatism or the cylindrical lens space is in the biplane, the distortion along or localization accuracy along the X and Y are about the same. So the localization precision are about the same along the XY throughout the whole depth. But because of the astigmatism introduced the distortion differently from X and Y location, the astigmatism will have localization precision very differently at different depths. This is inherent to distortion based, not to biplane based. So what we show here is in a densely labeled mitochondria and cytoskeleton, we will be able to image using the biplane multicolor image. We'll be able to investigate the interaction between cytoskeleton and the mitochondria. So this work received a new NIH award so that we can investigate the cell stress under the high glucose in animal models. So now let's move one step forward. We solved the challenge for spectroscopic analysis. Based on the spectroscopic analysis, we introduced the 3D imaging method by adding the biplane. But still, as I mentioned, and I showed the overall architecture again, For all the photons coming, we have to divide them into two groups for spatial localization and for spectral analysis. The downside is certain photons will be used for spatial imaging, certain photons will be used for spectral analysis. So we ask ourselves a question: can we use all photons for spatial localization, and at the same time, can we use all photons for spectroscopy analysis? So the solution is actually pretty smart. So one of my graduate students, he came up with this solution. It was super smart. So still the same grating, but this is a specially designed grating. This special design grating will have no zero order. So you look at the difference between A and the original schematic. There's a zero, there is a first. In the new design, there will be no zero. There will be a first, there's a first and a -1. So it has to be a specially designed. So what it gave us a 2 mirror image. Both of them are spectroscopy image. All right, these two are mirror image to each other. Both of them are spectral image. So what we do, if we flip them with respect to the center line, we can add photons to form, we can add all the photons to form a spectroscopy analysis. The center line itself will be the location of the molecule. So we call that virtual spatial image. We're not losing the spatial image, but we use all the photons for spectroscopy analysis. And these two photon groups collectively provide us the central location, and that's the virtual spatial image. What we gain out of this, we maximize the localization of all the photons. So this is the calibration process. On the left, these are single color calibration. So this is -1 order spectral image. This is +1 order spectral image. So you can see that they're actually mirror image. And we find the center line of this 2 mirror image and compare with the actual spatial image. They're actually highly overlapping. We calibrate for 2 color, and as long as we separate the label far enough, the 2 colors still work really well. So here's the demonstration. So we pick X47 and CF6880. And these two dyes are slightly far away, but with significant overlapping. So we pick this spectral window. And this is the demonstration of 2 color, fully utilized, symmetric dispersed spectroscopy single molecular analysis. It maximized the photon utilization for every single fluorescence emission. So now I show a 2D image. How can we go 3D? Again, we have 2 beams to begin with. Following the same principle, we add a pair of mirror as they are introducing additional optical path lengths. We have a biplane spectroscopy analysis using symmetric dispersed lens, symmetric dispersed system. So what we show are tracking of 3 quantum dots in 3D. So one of the interesting things I want to show, and then I'm sure many of you may aware, is when you buy a fluorescence label, you receive a menu that this is my absorption spectrum, this is my emission spectrum. There are 2 symmetric curves with very smooth signal. But in reality, if you are able to measure the fluorescence emission of the same molecule, of the same species from different molecule, they're actually not the same at all. They vary back and forth very much, can be as high as 40 nanometers. So I'm showing a Alexa Fluor 568. This is a rhodamine-based label, and you can see how large the variation is. If we zoom in into the spectroscopy analysis, you will see they actually shake back and forth from different molecules of the same species. This is called this is actually called spectral heterogeneity. It shows up not just from a single dye deposited on a glass substrate. It's actually happened almost everywhere in the localization microscopy if we can detect the spectrum. So what I show here are two microtubules, and you see that they're labeled by a rhodomy B, and they're so much different. Why that's the case? We hypothesize that it is caused by the local environment, caused the degree of freedom on the molecules. In this case, rhodomy molecules, because of rotation freedom and bending freedom. So we're trying to investigate how this freedom can be manipulated to our advantage. So I won't go into details of that. So if you recall, in one of the early slides that I show, the unwanted background is called fluorescence impurity. People did not pay attention to how to get rid of that until here, non-blinking semiconductor nanocrystal. It was published in 2009 on Nature. All right, great work. But if you look at that, retracted. Why that's the case? It was actually defluorescence impurity. So it was a terrible mistake. But I plot for these researchers. They followed the integrity, they retracted, and then they recognized that they made a mistake. So how to get rid of that? That's the question. Here, because we know the spectrum of each emission, assuming this is what we want, this is what we don't want, this is what we don't want. But by analyzing the emission spectrum of every emission, we will be able to tell this is what we want, this is what we don't want, just by looking at the emission spectrum itself. So we actually provided a different layer of freedom to reject things that we know that are not true. So this is demonstration. This is all the localization spots of molecules. We can use intensity filter to reject a majority of them. But if we apply the spectroscopic analysis to reject the unwanted impurity, quote unquote impurity, we can significantly reduce the number of localized reflects the true molecule clusters. So this is a demonstration. There are 1, 2, 3 clusters. And these are the first order spectral image. If we don't have the spectral image, 1, 2, 3 clusters, and get rid of all the background using intensity filter, we may say, okay, there's 1, there's 2, there's 3 clusters. But if we look everything carefully by analyzing their spectroscopy signatures, we'll be able to tell, no, these are all impurity. Not everything in this box comes from the true signal, and lots of them are actually impurity. Only the one at the bottom is the true signal. So it's provide a flexibility for us to reject the unwanted signal. So I would imagine if the researchers that I show who retracted their paper had a way to analyze their emission spectrum, they probably will have a better result. So by utilizing the spectroscopic analysis, we push this slightly forward to study the polymerisome. So polymerisome is a so polymerisome rely on the forms of nanocarrier can be used for a variety of drug delivery applications. One of the features for polymerisome is it has 2 ends. One is a polar end, one is a non-polar end. And if we use now red, which is interesting dye, is well, now red is combined with polar or non-polar, it's gonna show a blue shift of the fluorescence emission. So what does it mean? If the now red is floating in the substrate, it's gonna have a longer emission spectrum. As long as it's combined to the polymerisome, because of the polarity change, it will have a blue shift. So it provides us a way by analyzing the spectral signature of the emission, we will be able to count how many carriers, we will be able to estimate how big the carriers are with a much higher accuracy. So this is the process that I want to demonstrate. This is the image, a raw image of the polymersomes scattered on the substrate. These are signature spectrum information that are acquired from 1, 2, and 3 locations. These are mixture of polymersomes with specific binding null-red and with non-specific binding null-red. We have to select a band that with the highest affinity that show this is null-red binded with polymersome, and it can reject the unwanted background. So these are the results showing polymersome after rejection and the control image after rejection. To work with the image process, we develop a clustering algorithm that we call the Q-space. So the most used clustering algorithm is called dbScan or HDBScan. It's based on the intensity clustering, trying to calculate each emission localization with respect to the origin of the starting point and then calculate the weight. So we use part of the dbScan, but on top of the dbScan, we added spectroscopic analysis imposed onto the dbScan to name as a Q-space. It's actually increased the accuracy of the clustering a lot more. So we show here is, this is negative control, this is low concentration, this is high concentration. From the low and high concentration, we have PC means potential cluster by dbScan. And VC is validated cluster using Q-space or our technology. We show that if we don't know the fluorescence emission, we don't rely on the fluorescence emission to validate the clustering, we will have totally false readings. And we will have totally false readings in both concentration and size. Here we show a size estimate as comparing our Q-space with NTA. What is NTA? NTA is called nanosite. It's a technology, it's a commercial technology to count the concentration of nanocarrier. It's commercially available, but the limitation for the NTA is it can only accurately measure the concentration of nanocarrier within a very narrow range of concentration. When it's too high or too low, it just gave very false information. So we compare that. We compare our reading with the nanosite within the accurate range and validated our reading. Okay, so few more technology development here. So we saw the spectroscopy analysis. We introduced 3D image. We trying to improve the photon utilization efficiency, and then we're trying to improve the fluorescence impurity, trying to reject that. The holy grail for super resolution is how we can push the resolution even higher. Here's a concept, we call that regression, and I will show what exactly it is. So these are the point source function that we acquire. These are the localization based on the individual point source function localization. When we combine them, we have a new point source function, and this distribution corresponding to the uncertainty of the localization process, we can do this localization once again so that we can significantly improve the resolution. This is hard to picture. I'll show a simulation. So if we cluster 1, 5, 10, 20, 50, and 100, the localization accuracy will drop significantly, or in other words, the resolution will increase significantly. Why is that the case? So in other words, in localization microscopy, the precision is inverse proportional to the square root of photon count. So how to improve the photon count is the major challenge people are trying to overcome, either developing the bright dye or developing other method. So if we have a way to combine frames, remember, in order to achieve super resolution, we have to acquire thousands of frames. If we can combine frames, we can manipulate the square root of n by multiplied by nb's number of frames. So we can numerically increase the number of photons by a lot. This is nb is something we can control. So what we can show again here is, now this is a typical spectroscopy spatial image. This is a corresponding spectral image of quantum dot scattered on the substrate. These are 3 quantum dots. Quantum dot, as long as they have different size, they have different fluorescence emission. And the quantum dot, we love quantum dot for all the calibration study because they're so bright and they never bleach. So using one frame or using very limited frame, this is how they look like. By combining more frames, we will be able to separate them. So when we combine 100 frames, we will know from here, we don't know where they are. We know there are 3, but when we combine 100 frames, these are very well separated. There are 3 quantum dots so close to each other. The spatial separation is 6 nanometers. So this, again, this is not real. It's quantum dot. It's man-made object. How can we validate using the real object? We're lucky that people have built origami or nano rulers. What is that? So this is a DNA scaffold. DNA scaffold is built on single strand DNA and then bundle them. And the beauty of that is the nucleotide has a known size around 0.3 nanometer. So the spacing can be precisely controlled by how many nucleotides between the 2 labels. In this case, we buy the nano ruler with spacing of 23 nano, 11 nano, all the way to 6 nano. Although they are labeled by the same dye, but as I showed in the very beginning, the different molecule, even if they are from the same species, because of the local environment change, they will have slightly different fluorescence emission. In this case, they actually separate by about 15 nanometers. So we can utilize this spectral heterogeneity to separate them based on what I showed, spatial regression or PASER, in other words. We actually show that experimentally we can separate 5.6 nanometer spacing controlled by the number of nucleotides. So these are in 2D. You may ask, okay, these are in 1D. You may ask, how are we doing in 2D? So this is a nanograde. It's the same type of concept. It's a DNA origami. The spacing is controlled by the number of nucleotides. In this case, it's 10 nanometer spacing. This is confocal. This is the real storm without spectroscopy analysis. What we have is, with spectroscopy analysis, we add a 3rd domain. This is the lambda domain. Within the lambda domain, we cluster all the emissions and we can combine them. So that in this case, we are able to resolve the 7 out of the 9. The two of these did not generate sufficient number of fluorescence emissions, so we reject it. So we're almost down. One of my goal is trying to push this to the broad research field by other researchers, because I recognize that not everybody can build sophisticated optical system, and not everybody can write sophisticated algorithm to process all the data. So we just published a new paper to make this open source available. It's called Rainbow Storm. Rainbow indication that we do spectroscopy analysis. Storm is the acknowledged name for one of the family of localization microscopy. Rainbow Storm right now is online. We published not too long ago, and you can go to GitHub to find that. You can also go to my website to find that. This is some illustration. Rainbow Storm has 3 steps. First is calibration. calibration needs to be done before any data processing or data analysis. Calibration is system specific. We provide calibration for 2 implementations, both grating and prism, and we validate the data from published grating and published prism data. So I have to acknowledge the data sharing from the Berkeley group. And we feed the spectral calibration using either first order linear equation or second or third order polynomial, because prism is not linear in terms of the spectral dispersion. So then second step is the analysis. For example, if we want to acquire how they distribute in terms of spectral, how many photons per blinking, all the statistical signatures you will be able to acquire from Rainbow Storm. And the nice thing for Rainbow Storm is you don't have to install anything. It's a plugin for ImageJ. ImageJ is one of the most used open source software, highly recommended by NIH and by other nonprofit research organizations. So once the analysis is done, so for analysis, this is everything is in single color or in grayscale, and then you will get the distribution properties, the statistical properties of the labels that you acquire from your image. So the last step is visualization. So you load all the data you process, and then regardless how many colors are there, you can assign your own color, or the program will provide the default color. And you can adjust the color at your preference. So again, this is freely available online, and I welcome everyone to try it and provide feedback or tell me, oh, that's wrong, and you should do it this way. So one last topic. One of the major challenge for super resolution, as pointed out by Eric Benzik. So this is 2015 when he had a seminar at Berkeley. So I tried to, I wrote down what he said in the seminar. One of the major goals of the field, which nobody has been able to address really high resolution yet, is label-free imaging. In other words, can we do label-free single molecule localization microscopy? If it's true, that will be dream come true, because we don't need label, because everybody know that labeling is challenging. On the left, you will see a patent NU, stand for Northwestern University. This is a PMMA substrate with e-beam lithography on silicon substrate. So the feature size is 65 nanometers. And on top is the raw image per frame. At bottom, you are seeing the reconstruct as the frame is being accumulated. We did not apply any fluorescence emission. All that came from the intrinsic emission from PMMA. One more step. If you look at this, image, you see this pretty chaotic feature, but if you look at the image long enough, you will see that you see the grating pattern, the vertical strips from large spacing to short spacing. Again, this shows that we can create a feature with different feature sizes without doing any labeling. The PMMA after e-beam lithography provides signal for us to achieve super resolution image. This was interesting. We didn't know what was going on. And then we image the chromosome. So what is that? So we break a nucleotide. We break a cell nucleus, and we spread that on a substrate. Without doing any labeling, you will see the X-shape chromosome. And if, after all the reconstruction, you see the X-shape very nicely, and you even see the slightly defragmentation of the chromosome. What's more fun is if we don't break the cell nuclear membrane, just image the whole nucleus after fixation without labeling, you will see the slightly decay, so this eventually bleached out. And once we finish the localization microscopy reconstruction, you see a chaotic image. It's not so chaotic when you zoom in. So if we zoom in this red square, this is what you see. Still slightly chaotic. Let's zoom in even more. And now you see the features. If we color code the feature by the concentration of nucleotide, in this case, it's a CT domain. We actually had a very interesting image of the nucleotide, of the nucleus with the nucleotide concentration distribution. So this is not something really fake. We're actually doing the nucleotide imaging without label in the whole intact nucleus. So this work received the 2017 National Science Foundation, National Academy of Science Award, as we first report the observation of the autofluorescence from the nucleotide or from DNA. So I'll stop here. Because of the time constraint, I have a lot more can share if we have more chance, future opportunities to share. I have to acknowledge the great help that I have received from my collaborators, Dr. Nader Shibani from Wisconsin, Dr. Zhang, Dr. Beckman, Dr. Bao, Dr. Scott, Dr. Shears, Shilafi from Northwestern, and Dr. Ying from Buffalo. We do lots of machine learning aspect, and then Dr. Pablo from University of Chicago. We are studying the modeling of the copolymer as the phase change of the copolymer modeling to investigate the methylation and the chromatin organization from the imaging data. These are my very talented group, half of my group that are focusing on super resolution microscopy. I receive funding support from National Science Foundation and National Institute of Science, National Institute of Health, I'm sorry. I form a company, but not for super resolution, it's for eye imaging. This is the bird eye view picture of Northwestern campus. It's not so big, but it's in a good size. We have a small lake inside the campus, and this is Lake Michigan. It's like the sea. This is where I typically go for a walk in a good day. My office is somewhere over here, and I'll stop here. I'd like to take any question that you might have. Thank you.
Okay, thank you, Professor Zhang, for the wonderful presentation on the technical development and applications of